everyone. This is the fifth lecture for your chapter Respiration in Plants. Till now, we have studied glycolysis, which was the first pathway and the most common pathway in all the living things, right? In which we saw partial oxidation of glucose. So the glucose was getting converted into two molecules of pyruvate. Along with this reaction, we were getting two molecules of ATP and two molecules of NADH. This pyruvate enters the mitochondria and undergoes the link reaction. Okay, and in this link reaction, pyruvate is converted into acetyl CoA. During this reaction, one molecule of carbon dioxide is released and one molecule of NADH. Moving ahead, this acetyl CoA enters the Krebs cycle or TCA cycle. In this citric acid cycle, more and more NADH as well as FADH2 are generated. We saw in the last class how NADH and FADH2 they transfer their electrons, okay, and these electrons they are transported through different complexes of the electron transport chain or electron transport system. After the last electron transport complex, okay, the last complex, which is complex number 4, that complex finally gives the electron pair to oxygen, okay, and this oxygen with the electron pair as well as two protons, it generates one molecule of H2O or water. So till here we have seen, we also saw the chemiosmosis hypothesis, okay, and how ATP is exactly generated. So till here there is nothing, I don't think there should be any doubt, okay. So one thing which you must have missed during this is what happens to the NADH that is present in the cytoplasm. Now all of these NADH, okay, during this pyruvate dehydrogenase complex reaction or Krebs cycle, these all NADH they are generated within the mitochondria, right? And so it is easier for these NADH and FADH2 to directly go to the electron transport chain, right? Now what happens to the NADH which was actually generated outside the mitochondria, okay, into the cytoplasm? So we will be talking about this NADH molecule today. In the second portion of the lectures, today's lecture, we shall be also discussing the net ATP gain from one molecule of glucose as well as the percentage efficiency of this oxidative cellular respiration. Clear? Let us understand the whole process a little bit better. For that, for this one molecule of NADH which is generated outside the mitochondria or in the cytoplasm, what happens to it? So for that, we have to study shuttle systems. Now, what is a shuttle? So, if you know or if you have seen sports, if you know this in badminton, they use this shuttlecock, right? So, why is it called shuttle? Basically, as you can see, it can go back and forth, okay, from one player to another and then back again to the first player. So, when anything is moving ahead and coming back, okay, so it is going back and forth, we call it shuttling. Same thing if you know in weaving, okay, how clothes are made. So in that also you can see this lady, she has this one instrument, okay, which is passing through and coming back, right? So this instrument actually is also known as a shuttle. In common terms also you must have used, or common day-to-day life also you must have heard shuttle where in case of rickshaws, right? So you have shuttle rickshaws which let us say they work on certain route, from Crystal Mall to KKV Chowk and from KKV Chowk to Crystal Mall. So these you call shuttle, okay? You give them 10 rupees, 20 rupees and they will bring you from point A to point B, okay? So this way shuttle is basically something which is bringing a thing back and forth, right? After understanding the meaning of shuttle, okay, now I hope it has been imprinted on your brain that what a shuttle means. Let us understand, okay. So what happens to the NADH generated during glycolysis in the cytoplasm? Okay, during glycolysis there are two NADH molecules produced per molecule of glucose. So in case of anaerobic respiration, that is when there is absence of oxygen, then fermentation will take place, that you already know. So if alcohol fermentation takes place, which means there is involvement of enzyme called alcohol dehydrogenase, then this enzyme or that alcohol fermentation pathway will utilize this NADH. Okay, it will donate its hydrogen as well as so the proton as well as the electron to your alcohol. Okay, it will give it to aldehyde. Aldehyde will get converted into alcohol, and NAD plus will be free. Same thing if it is lactic acid fermentation, then in the pyruvate, it accepts one proton, it accepts one electron, and frees the NAD plus. Okay, from NADH it becomes NAD plus, and we have already seen the reaction, and the lactic acid is produced. Right. So in case of anaerobic respiration, this is what happens to the NADH. In case of aerobic respiration, we have these shuttle systems. Okay. So shuttle systems, they can get NADH across the impermeable mitochondrial inner membrane. Now I want you to recall the structure and the functions of the mitochondrial membranes. Okay. If you remember the outer membrane, it has porins. Okay. It is very porous. It allows easy transport of things across it. So things can easily go inside and go out of the outer mitochondrial membrane. However, the inner mitochondrial membrane is very strict. Okay, it is very strict. It will only permit some things to pass through it. Right. So it is mostly impermeable for NADH. So this NADH from the cytoplasm, it cannot enter the mitochondria, and from mitochondria, NADH cannot easily leave the mitochondria. Clear? So for this, to transport this NADH, okay, or rather the electrons and the protons from NADH to mitochondria, we have shuttle systems. As you can see, I said shuttle systems, not shuttle system, which means there are two of them. One is known as malate aspartate shuttle, and the other one is called glycerol phosphate shuttle. Let us begin with the first one, which is malate aspartate shuttle. Just a very quick recap where you have heard malate. So you have heard malate in C4 pathway. You have heard of malate in CAM pathway, as well as in carboxylic acid cycle, right? Tricarboxylic acid cycle, TCA cycle. Fine. Where have you heard of aspartate? So aspartate is actually an amino acid, right? And this amino acid we have studied in biomolecules, the structure. If you have any doubt or if you do not remember exactly, I would recommend pause this lecture here and go back and refer to its structure, okay, and what kind of amino acid it is. Fine. Chalo. Moving ahead, let us start with the malate aspartate shuttle. Where is this type of shuttle seen? So this shuttle or this pathway is seen in heart, in liver and kidneys. So as you can tell from the statement itself, it is seen only in animals. Clear? So it is seen in the heart, it is seen in the liver and in kidneys. It is a reversible shuttle. Reversible in the sense that the NADH can be transferred from outside the mitochondria to inside, and the opposite is also possible. It works according to the NADH concentration gradient. Whenever there is this square bracket, I hope you understand that I'm talking about concentration. So according to the NADH concentration gradient, the pathway will or the shuttle will work. If the NADH concentration in mitochondria is high, then from inside to outside. And if NADH concentration outside is high, then outside to inside. Clear? So it is reversible. Here, there are two main antiporters. Now, I know I have not yet taught you this term, but I just want you to know that antiporters, anti means opposite, okay? And porter. Have you ever heard of coolie, right? On the railway stations and all, you have coolie. So in English, you call them porter, right? So porters are nothing but they are something which carry, okay? Somebody who carries stuff. carries your luggage, carries all the burden, all the weight. So here from the name itself, you can come to know anti-porters do what? They do this lane देन. Okay, in Hindi or in Gujarati, if we say, it is basically एक हाथ से ले, एक हाथ से दे. Okay, so this is called anti-porter. Basically, they will transport one molecule, any one molecule, let us say A, from outside to inside, and at the same time, it will transport a molecule, let us say B, from inside to outside, right? So, ek haath se le, ek haath se de are called antiporters. So, these antiporters, there are two types which play an important role. The first one is malate alpha ketoglutarate carrier, and the second one is called glutamate aspartate carrier. Now, do not get worried from the name itself. Let us just see the pathway, okay? See the shuttle. We will begin with, let us say here, okay? Outside. So, outside you have this oxaloacetate in the cytosol, okay? This membrane here is showing the inner mitochondrial membrane. Now, you must be wondering why across the inner mitochondrial membrane there is cytosol and matrix. Remember, I told you that mitochondrial outer membrane is permissible, okay? It is open for every molecule to pass easily. So we are not considering the outer membrane at all, because the molecules can easily pass through the outer membrane. So what we are looking at? We are looking at the cytosol and we are looking at the matrix, and in between we have the inner mitochondrial membrane. Clear? Now you have a molecule of oxaloacetate outside. Oxaloacetate, as you know, is a 4-carbon organic compound, okay, organic acid. Now this oxaloacetate will be converted into malate. Clear? So oxaloacetate to malate, this is the first reaction. The reaction is catalyzed by enzyme known as malate dehydrogenase. Okay, malate dehydrogenase, we have heard of this enzyme in TCA cycle, right? So the same TCA cycle enzyme is also found in the cytosol or your cytoplasm. Clear? So in the cytoplasm, what is this malate dehydrogenase doing? It is doing the opposite reaction. Okay, it is converting oxaloacetate into malate. And as you can see, it is utilizing that NADH. Remember, we are talking about the NADH which is produced during glycolysis. So it is present in the cytosol. Now we want to transport this NADH into the mitochondria. So this is how it happens. This NADH, okay, it will transfer its hydrogen, so H, okay, in form of H plus, as well as the electrons, okay, along with this H plus. So in total, the whole hydrogen atom. This hydrogen atom will be donated to the oxaloacetate, and this will produce malate. So now you can see malate actually has that electron as well as proton. This NADH will get converted into NAD plus, and this NAD plus is free to continue carrying forward the glycolysis reaction, right? Okay, so oxaloacetate to malate, the enzyme is malate dehydrogenase, and one molecule of NADH is used, okay, to reduce oxaloacetate. Now this malate, okay, there is a transporter on this inner mitochondrial membrane, which is known as malate alpha ketoglutarate carrier, right? So remember, this is the first antiporter we talked about. So what will this molecule or this protein do? It will transport this malate from cytosol into the mitochondria. Clear? And along with malate, what is it doing? It is transporting alpha ketoglutarate. Now alpha ketoglutarate also we have studied in TCA cycle. So alpha ketoglutarate from the matrix of mitochondria, it will get transported outside into the cytosol. So I told you ek haath se le, ek haath se de. So from one side it is transporting malate from outside to inside, and alpha ketoglutarate from inside to outside. Clear? So malate alpha ketoglutarate carrier. Now the third part. What will happen now? The malate, this is the normal TCA cycle reaction. That malate will get converted into oxaloacetate by an enzyme called malate dehydrogenase. Okay, this is the last step in TCA cycle where you are regenerating the oxaloacetate, right? And during this reaction, you are getting one molecule of NADH. Now, if you can see the outside NADH, okay, it's electron and proton, they are transferred and now they are inside, okay. So this NADH now has those electrons as well as the proton. Clear? So this is how the NADH is transported from outside to inside, not directly, but through shuttle. Fine. Now let us see what happens to this oxaloacetate now. This oxaloacetate gets converted into aspartate. Aspartate is a 4-carbon amino acid. Okay. How 4 carbons? It has one alpha carboxylic group, alpha amino group. This is the alpha carbon. And in its R chain, it has CH2COO minus. Okay, it is an acidic amino acid. So oxaloacetate, it gets converted into aspartate. Now, if you see the difference is presence of this amino group, right? NH3 plus. Who is donating this amino group? So glutamate, another amino acid. Okay, glutamate is same as aspartate except it has one more CH2 in its R group. So this glutamate will transfer its ammonia, okay, or NH3 plus group, ammonium group, to oxaloacetate, and this will get converted into aspartate. After donating that ammonium group, glutamate gets converted into alpha ketoglutarate. Clear? So now, what do you have after the fourth reaction? You have aspartate and you have glutamate, right? So remember, the second carrier, the second antiporter was glutamate aspartate carrier. What will this glutamate aspartate carrier do? It will transfer this aspartate, okay, outside from matrix to cytosol. So now you have the aspartate out in the cytosol. On the other hand, it will take glutamate from the cytosol and transfer it into the matrix. So again, ek haath se le, ek haath se de. Aspartate is going out, glutamate is coming in. Fine. This aspartate, again, there is an enzyme, same enzyme, aspartate aminotransferase. What will it do? It will do the opposite function. From aspartate, it will take the amino group, give it to alpha ketoglutarate. Once alpha ketoglutarate receives this amino group, it gets converted into glutamate. And aspartate gets converted into oxaloacetate. And the shuttle is ready to work again. Clear? So as you can see, the reactions are same on the both sides. They are just moving in the opposite direction. Right? Now, why is this required? Okay, why are we sending this aspartate out? And why are we bringing that malate in? So malate, do you understand that we are bringing malate inside because it has that hydrogen, right? Now, why we are pushing aspartate out is because this malate, okay, it cannot, now after donating hydrogen, this oxaloacetate cannot leave. So it will get converted into some molecule which can leave the mitochondrial membrane, right? So which is aspartate. Once aspartate goes out, it again gets converted into oxaloacetate, and oxaloacetate gets converted into malate, and the cycle can keep on continuing. Clear? So there are just two main reactions you have to remember: oxaloacetate to malate. Clear? And this is just a simple reaction of reduction. Okay. Then the other reaction which is important is alpha ketoglutarate to glutamate, and during this process there is aminotransferase, or the reaction is known as transamination. Transamination meaning from one molecule to other molecule you are transferring the amino group. Clear? So from let us say glutamate, you are transferring it to oxaloacetate, right? Then it will become aspartate. And if from aspartate you are transferring the amino group to alpha ketoglutarate, it becomes glutamate. Clear? So this is the malate aspartate shuttle, in which basically what is the net reaction? Net, you are getting the outside NADH is getting transported inside. Okay, not directly but through the shuttle system. Now let us study the second shuttle system. This is known as glycerol phosphate shuttle. Okay, now this is a little simpler compared to the previous shuttle. Let us study it. It is seen in the brain, in the skeletal muscle, insect flight muscle, and in all plants. Clear? So if somebody talks about plants, glycerol phosphate shuttle is seen in all plants. In case of animals, in brain, okay, in your brain tissue, in your skeletal muscles or striated muscles or voluntary muscle, whatever you want to call it, right? So in these muscles, then in insect flight muscle. So if you see, let us say a butterfly, and the butterfly is able to flap its wing and fly, so it has these muscles, okay, which help it flap the wings. These muscles are known as insect flight muscles, okay, the muscles which are helping it fly. They have glycerol phosphate shuttle, and all plants have glycerol phosphate shuttle. Unlike the previous shuttle, that is malate aspartate shuttle, glycerol phosphate shuttle is irreversible. You cannot reverse it, okay, it can only move in one direction. The NADH will be transported, the electrons or protons from NADH in cytosol will be transported towards mitochondria only. It cannot come back. It is effective even when the cytosolic NADH concentration is low. So even though only few molecules of NADH are produced during glycolysis in the cytosol, this pathway or this shuttle will be working, okay, and you will get every NADH converted into ATP. Shuttle systems are absent in prokaryote. Now this is my homework to you. Find out why in prokaryotes you do not require shuttle systems. The answer is very very simple. Okay, chalo. Now let us study this glycerol phosphate shuttle. Same way as previous diagram. Here on the left hand side you have cytosol or your cytoplasm. In between you have the inner mitochondrial membrane. Outer is not mentioned because all these molecules can easily pass through the outer membrane. Okay, and then on the right hand side you have the mitochondrial matrix. And this is what is happening. You have this NADH outside. Okay, this is the cytosolic NADH that you got from glycolysis. This NADH will be used to reduce dihydroxyacetone phosphate. DHAP or dihydroxyacetone phosphate we have studied in case of glycolysis, and glycolysis takes place where? In the cytosol, right? So this is the structure of DHAP, okay, which is nothing but CH2OH at the top, C double bond O, because it is a keto triose sugar, right? So it has a ketone group, and at the bottom it has CH2O, and instead of one H, the phosphate group is placed. So it becomes dihydroxyacetone phosphate. So this is the molecule. Now this NADH, okay, will be utilized to reduce this DHAP. The enzyme which catalyzes this reaction is known as glycerol phosphate dehydrogenase. Okay, and what will it do? It will basically add hydrogen to this molecule. When hydrogen is added, it gets converted into glycerol 3 phosphate. Okay, everything is same except in this area, okay, where there is C double bond O, you now have this hydrogen which is attached. Clear? So reduction of dihydroxyacetone phosphate gives you glycerol 3 phosphate. Very easy way to remember this is in I think last or second last lecture only I had told you that when you uh, reduce, okay, when you reduce carboxylic acid, you get acetaldehyde, aldehyde, okay. From carboxylic acid, when you reduce it, you get aldehyde or ketone, and when you reduce aldehyde or ketone, you get alcohol, right? So the same thing is happening here. You have ketone group, and this ketone group is getting reduced, so you're getting alcohol. This alcohol, this 3 carbon alcohol, is known as glycerol. Okay, so glycerol 3 phosphate. On the third carbon it has this phosphate group, so glycerol 3 phosphate. Now this glycerol 3 phosphate, there is another enzyme which is known as flavoprotein dehydrogenase. This enzyme is present on the inner mitochondrial membrane. What is this enzyme doing? It is basically converting FAD, okay, which is a flavin adenine dinucleotide, into FADH2. into its reduced form. Where is it getting this hydrogen from? The hydrogen is it is getting from glycerol 3 phosphate. So glycerol 3 phosphate plus FAD catalyzed by flavoprotein dehydrogenase will give you FADH2, and you will regenerate the dihydroxyacetone phosphate. Clear? Now this FADH2, because it is directly present in the inner mitochondrial membrane, this FADH2 will be directly given to your electron transport chain or electron transport system. Okay? The electrons will be passed through all the complexes, finally given to oxygen, and during this process, whatever protons will be pumped out will be used by the ATP synthase to generate ATP. Clear? So one thing to remember in this shuttle, NADH is getting converted, or the electron from the NADH is getting converted into electron of FADH2. Now FADH2, we know, generates less ATP compared to the NADH. Clear? So here in glycerol phosphate shuttle, because NADH is converted into FADH2, we generate few number of ATP less. How many exactly? We shall study in the next slides, okay, in coming slides. So is this much clear? In malate aspartate shuttle, you are converting NADH, okay, the electrons from NADH are going to the electrons in NADH inside the mitochondria. However, here in glycerol phosphate shuttle, the NADH, okay, electrons from the NADH are getting transferred to FADH2, okay, and so there will be a difference in the ATP or the number of ATP generated here. Clear? You will understand this better when we discuss the ATP gains. Clear? Now the number of ATP molecules synthesized depends on the electron donor, okay, who is donating these electrons. It depends on that how many number of ATP are generated. Who is the electron donor here? So we have only two. We have either NADH or FADH2. So oxidation of one NADH. Oxidation of NADH meaning what? NADH giving you NAD plus H plus electrons. So this process is called oxidation. Okay. So oxidation of one NADH gives you 3 ATP, and oxidation of one FADH2 gives you 2 ATP. So from this, can you understand that if glycerol phosphate shuttle is used to transport the electrons from NADH into the mitochondria, then we will generate 1 ATP less. Okay, chala. Now, apart from the electron donor, it also depends on the shuttle used. If glycerol phosphate shuttle is used, it gives you 1 ATP less per NADH present in the cytosol. And if you use malate aspartate shuttle, you get full 3 ATP per NADH of your cytosol. Now, the theoretical calculations of ATP gain can be made only under certain assumptions. Okay, you cannot just directly calculate. You have to make some assumptions to make sure that you get proper results. What are these assumptions? So the first one, assumption or precondition. Okay, it is so first assumption or precondition is only glucose is being respired. Okay, we saw respiratory substrates in the first lecture itself, and there could be many. So here we are assuming that only glucose is being respired. No other alternative substrates are entering in the pathway at any stage. Okay, so only glucose is being respired and nothing else is entering the pathway at any point. Second, there is a sequential orderly pathway functioning. Sequential matlab step by step, right? An orderly, very ordered, very disciplined pathway functioning. What does this mean? So if a glucose is entering, it will undergo glycolysis. After glycolysis will be link reaction followed by TCA cycle, then electron transport system, oxidative phosphorylation. Clear? So there is no gadbad in this sequence. It is going in the sequential order. Clear? No intermediates are used to synthesize other compounds. So there are many intermediates in the TCA cycle as well as in the glycolysis. So these intermediates are not used to make something else, okay, during this calculation. NADH synthesized in glycolysis is transported to mitochondria and undergoes oxidative phosphorylation. Yeah. So the NADH that you are getting in glycolysis, okay, which is present in the cytosol, we are assuming that all of these NADH, they are getting transported into the mitochondria. Clear? And after entering the mitochondria, they undergo the electron transport system as well as oxidative phosphorylation. Now, of course, these are all theoretical calculation. In real life, practically, these assumptions are invalid. Okay, in a living system, these assumptions are invalid. Here, in living system, all the pathways, they work simultaneously. Substrates, they can enter pathway at any time and are also withdrawn at any time. Okay, whenever necessary. Also, not all NADH produced during glycolysis, okay, present in the cytosol, will be transported into the mitochondria during normal living conditions. But just for our theoretical calculation, we have assumed all of these, and now we shall see. Okay. So now there is a practical gain versus theoretical gain. Practical gain is like we will calculate right now, okay, and theoretical gain is what you have to remember for your exams. This is just for your knowledge, and theoretical gain is for your remembering, okay, for your use, practical use. Okay. So for NADH, 10 protons are pumped out per pair of electron transferred to oxygen. Okay, so if you have one NADH molecule, right from complex 1, then complex 3 and complex 4, all of these three complexes in total, they release 10 protons, okay, from mitochondrial matrix into the intermembrane space. So you can say 10 protons per pair of electron. Fine. Now we had also seen when we discussed the ATP synthase that when 4 protons flow back, okay, when 4 protons come back from the intermembrane space into the matrix, one molecule of ATP is generated by the ATP synthase. Okay. So now from this, can you find out? You have 10 proton which are pumped out per NADH molecule, and per ATP molecule 4 protons are coming back. So to find out how many ATP are generated per molecule of NADH, you just divide 10 by 4. So 10 by 4 is 2.5. So per molecule of NADH, 2.5 molecules of ATP should be produced. Same way, calculating about FADH2. FADH2, the complex number 2, okay, succinate dehydrogenase complex. Now this complex actually does not pump out any proton. Okay, then complex 3 and complex 4, they pump out in total 6 protons from matrix into the intermembrane space. So same way, 4 protons are required for generating one molecule of ATP. So 6 by 4, okay, will give you 1.5 ATP. So this is the practical gain. Fine. Per molecule of FADH2, you should get 1.5 ATP. When you will study in higher standards or in your further studies, okay, higher studies, if by chance you come across this topic again, in higher studies you will be considering 2 ATP and 1 ATP respectively. Fine. But for your NCERT, they are rounding off these figures. Okay, rounding off these figures, you get the theoretical ATP gain, which is nothing but 3 ATP per NADH and 2 ATP per FADH2. So from 2.5 it becomes 3 and 1.5 becomes 2. Clear? So these are the values you have to remember. But I had lot of questions from students previously. They used to ask that how do you decide how many ATP are generated per molecule of NADH, right? So this is the calculation, how you can actually calculate, okay? Fine. So remember this much, 3 ATP per NADH molecule and 2 ATP per FADH2 molecule. Chalo. This is the whole summary, okay? Now you are going to calculate the final number of ATP. Ready? Chalo. Along with me. We start with the first pathway, which is glycolysis. In glycolysis, there is a reaction where 1,3-bis phosphoglycerate, okay, 1,3-bisphosphoglycerate is getting converted into 3-phosphoglycerate. At this point, you are getting 1 ATP, right? Remember this reaction, it will be proceeding in double, okay? Because from one molecule of glucose, you are getting 2 molecules of bisphosphoglycerate, right? So, 1 molecule of bisphosphoglycerate getting converted into 3-phosphoglycerate will give you 1 ATP. This is an example of substrate level phosphorylation. Then again, at the last step of glycolysis, phosphoenol pyruvate is getting converted into pyruvate. This reaction is also giving you 1 molecule of ATP. So together, how many ATP you are getting? You are getting 4 ATP, right? But why I have not written 4 is because during glycolysis reaction, we are also investing 2 ATP, right? So net ATP gain is just 2. Hence, I have not mentioned all of these in multiplied by 2. Clear? Fine. Now, phosphoglyceraldehyde, okay, PGAL, phosphoglyceraldehyde, to bisphosphoglycerate. This reaction is generating 2 molecules of NADH, okay, 1 molecule per reaction. So, in total, there are 2 NADH synthesized in cytoplasm. Depending on the shuttle, okay, if they use glycerol phosphate shuttle, then you will generate 4 ATP per molecule of, 4 ATP in total, okay. And if they use your malate aspartate shuttle, then you will get 6 ATP, which means 3 ATP per molecule of NADH. Clear? Fine. So this sums up the glycolysis. You have 1 ATP, 1 plus 4 or plus 6. We shall see. Then you have the link reaction. Link reaction is the reaction which will transfer your pyruvate into the mitochondria, okay, and convert it into acetyl-CoA. So this reaction is carried out by pyruvate dehydrogenase. Pyruvate is converted into acetyl-CoA, and during this process, okay, per reaction you are getting 1 molecule of NADH. There are 2 pyruvate molecules, so you are getting 2 NADH. These 2 NADH, because they are present directly in the mitochondria, they will give you 6 ATP, okay, 3 ATP per molecule of NADH. Then isocitrate, okay, isocitrate dehydrogenase enzyme will give you alpha ketoglutarate. At this point also you are getting 2 molecules of NADH and hence 6 ATP. Alpha ketoglutarate, again it will undergo oxidative decarboxylation, and during this process alpha Kg is converted into succinyl-CoA. This reaction also is giving you 2 NADH and hence 6 ATP. Now, of course, all of these ATPs you are not getting directly at the Krebs cycle level, you are getting it through oxidative phosphorylation. Yeah? Fine. Now, there is one reaction in the TCA cycle where you are getting ATP or rather GTP. Okay, but I had told you that GTP is equivalent to ATP. So, this reaction is from succinyl-CoA, you are generating succinate. And in this reaction, you are getting 1 ATP or rather 1 GTP per reaction. So in total 2 reactions, okay, so 2 ATP, and this is an example of substrate level phosphorylation. Then you have succinate to fumarate. The enzyme is succinate dehydrogenase, and you know this is a special enzyme, right? This enzyme is actually giving you 2 FADH2 molecules. This FADH2 will give you 4 ATP in total, okay, 2 ATP per molecule of FADH2. And last dehydrogenase is malate dehydrogenase. Malate will give you oxaloacetate and 2 molecules of NADH. These 2 molecules of NADH will in total give you 6 ATP. Now just add up all of these ATP numbers, fine, and you will get 36 or 38 ATP in total, right? If glycerol phosphate shuttle is being used, then you are getting 36 ATP. If malate aspartate shuttle is being used, you will get 38 ATP. Clear? So this is the only difference. However, I think in your textbook, according to your textbook, 38 is the better answer. Again, I don't remember actually. Just have a look, okay, in your textbook. Just find out if 36 is given or 38 is given. whatever is mentioned in your textbook in the NCERT syllabus, that number will be considered. Fine. Chalo. Now the final slide or the final topic for today's lecture, finding out the efficiency of aerobic respiration. Remember, we had found out efficiency of anaerobic respiration as well, and we found out that it is only 2% efficient. Okay, so from one whole molecule of glucose, we are only getting 2% of its energy. Now let us find out how efficient aerobic respiration is. So here again with the same calculations, burning one mole of glucose, okay, will give you minus 686 kilocalories per mole. Okay, so per mole of glucose you are getting minus 686 kilocalories. Minus, I told you, it is because it is an exergonic reaction. Okay, energy is given out. Fine, so 686 kilocalories. Now I had also told you this, that when the ATP, okay, it breaks, or the ADP and this phosphate bond, it breaks, it releases minus 8 kilocalories per mole of ATP. Clear? So per mole of ATP you're getting minus 8 kilocalories. Now aerobic respiration, okay, of 1 mole of glucose is giving you 36 ATP. We have just seen it now, right? We just calculated the whole um, uh, all the ATP gains, and we found out that 36 ATP or 38 ATP are produced from 1 molecule of 1 mole of glucose, right? 1 mole will give you 36 mole of ATP here. Fine. So just multiply 36 into 8 will give you 288. Okay, so 288 kilocalories you are getting per mole of glucose by aerobic respiration. Now originally how much you should get? You should get 686, but you're getting only 288, right? So what is the difference? What is the efficiency? 288, which is the realized value or the actual value, actual energy you are getting, divided by the original energy you should get from 1 molecule of glucose. 288 divided by 686 and multiplied by 100, because you want to find out percentage efficiency. When you put these numbers into the calculator, you will find out it is only 42% efficient. Okay, so the remaining energy, okay, out of 100% energy, out of the 686 kilocalories, only 288 kilocalories, okay, 288 kilocalories are present in form of ATP. The rest energy is lost in form of heat. And here remember, I had discussed the second law of thermodynamics, right? So here we are not what you can say breaking the second law of thermodynamics. Okay, all of this extra energy will be lost in form of heat at each step of the reaction. So that was all about today's lecture. We will see you tomorrow. Okay, so see you next time.